Hi, everyone. Thank you for joining today. We'll get started in about 5 minutes on the hour. Alrighty, we'll go ahead and get started. Hello, everyone, and thank you for joining today. Welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from the series are made available on our YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled A Practical Approach to Integrating eDNA into Stock Assessment in Fisheries Management Advice by Baetscher. Diana leads the environmental DNA research at the Alaska Fisheries Science Center in Juneau, Alaska. Her projects apply genetic methods to a range of ecological fisheries and management questions, including population genetics, rockfishes, and seabirds, and foraging ecology diets in northern fur seals. Diana started her role at the Alaska Fisheries Science Center in 2021. Thank you, Diana, for presenting today. And it looks like we can see your slides, so I will now pass it off to you.
Thanks for that intro, Nicole. It's great to be with everyone remotely today. And I'm going to be talking about some collaborative work that I started almost exactly a year ago about integrating eDNA into stock assessments. And you can see there are a number of other folks who have been involved in this. So this project began with what is sort of a simple question, but Dan Gaethel, a stock assessment scientist at the Alaska Fisheries Science Center, asked me, is there any benefit to having eDNA on a survey? And this was sort of surprising because I feel like for over a decade, the eDNA community has been talking about the utility of eDNA for fisheries applications. And simultaneously, there's this need to provide lower cost, more efficient, sort of next gen type surveys. So relying less on pulling nets and more on some of these novel technologies that will allow us to be more efficient with our limited resources. And the promise of eDNA has been that we can do faster sampling at lower cost, and that it should be relatively easy. And I, of course, use easy in quotes because those of us who have either done sampling at sea and or generated and analyzed data know that that it's not necessarily as easy as sometimes we claim that it might be. And that Dan asking this question sort of highlighted this lack of communication or lack of understanding between fisheries scientists and geneticists. So although we're publishing in our journals as geneticists and talking about the utility of eDNA for these very clear fisheries applications, that communication is not necessarily being seen or understood by the folks who are actually helping manage the fisheries or providing management advice to fisheries councils. And as Dan and I began digging into this, we realized that there was this limited overlap between our disciplines and pretty limited overlap in expertise. So eDNA researchers, we collect and generate data. The stock assessment scientists, they build their stock assessment models. And somehow the data that we generate is supposed to be useful to stock assessment scientists. But neither of us really could describe how it was going to be useful. And part of that problem, and here I'm happy to sort of say that these things are true for me, and they're probably not true for every eDNA researcher, but but I was mostly unfamiliar with stock assessments, what the components were, how they were put together, how the models actually functioned. And I would argue, based on what I've read in the eDNA literature, I think that's more broadly true, that eDNA researchers don't have a lot of familiarity with stock assessments and don't have a lot of familiarity with the type of modeling that goes into the assessments. And simultaneously, sort of on the other side of things, the stock assessment scientists have very little familiarity with eDNA terminology and data. And sort of further building these walls or these silos, we have jargon, like a lot of jargon. So eDNA researchers talk about primers and the importance of primer choice. We talk about quantitative PCR and compare that to digital droplet PCR and compare that to metabarcoding. We use bioinformatics. And then we show plots of our results that look sort of like this bar plot and talk about how that may or may not actually represent the true proportions of what we're sampling. So all of that is actually, sort of very much siloed within our research. And the stock assessment scientists, meanwhile, talk about indices of abundance, selectivity, catchability, availability, and uncertainty. And all of those things are technical terms with actual definitions, of course. And then this lovely equation I just actually pulled out of the Pacific Hake stock assessment. And that likelihood equation is the type of equation that is sort of commonplace in those documents. So we're fundamentally thinking about data in different ways, and we don't even necessarily have the terms to communicate across disciplines. So what that led us to realize is that we actually need to be very explicit about building interdisciplinary collaborations for management goals. So if we want eDNA to be useful, it needs to be part of an explicit design, and it's not just going to happen organically. And I want to pause here because there were some important things happening from our NOAA colleagues sort of at the same time and even a little bit earlier. So this, in my mind, is somewhat of a seminal paper by Ole Shelton and others from the Northwest Fisheries Science Center. And what Ole and others did was generate quantitative estimates of Pacific hake along the west coast of North America. So in this figure, in panel A, you see the stations, and they really do a ton of sampling along the west coast, and actually even more because it's international, so it goes into Canadian waters. And then in the middle panel, what Ole did was generate an index based on the quantity of DNA in the water samples. And so the heat map shows, the relative amount of eDNA along the, the sampling space. And then the final panel shows the acoustic biomass from those same sampling sites. So you can see that, although the DNA and the biomass might not be perfectly analogous, there's a pretty nice correlation in some of those hot spots. And one of the other things that Ole did in this paper is developed methods to account for the process and sampling uncertainty associated with the eDNA collections. So, he's essentially using the same types of approaches that the assessment scientists and that the fisheries scientists are expecting when they are using data streams for assessments. And then the final thing that I think is really notable about this paper is that it actually is using the spatial scale of the assessment. So, that's a multi-thousand kilometer survey grid, and they managed to collect eDNA samples from the whole thing. So it's directly comparable in terms of the scale of the assessment. So you have this, sort of correlation between the eDNA concentration and the acoustic biomass. And where you see differences, one can think about it in terms of the difference in gear and the selectivity of that gear type. So again, translating some of our eDNA results into terms that are understandable by the fisheries science community. Okay, so given that, what we thought about doing was getting the folks from the Northwest Center who were involved in that hake work and the folks at the Alaska Fisheries Science Center who were interested in how eDNA could actually be leveraged in their assessments, and writing a paper, describing how do we do this. And we had a couple of false starts because it turns out that jargon and disciplinary silos were pretty challenging to overcome initially. But we have a paper right now in review at Fish and Fisheries, and I wanted to highlight that we're putting it in Fish and Fisheries, hopefully, so that it can be read by the fisheries scientists and the assessment scientists. If we put it in environmental DNA or molecular ecology or a genetics journal, it's not going to be seen by the same people as if we're, sort of targeting an audience in the fisheries science world. So that was a pretty explicit choice. And so what does this roadmap entail? What we have is this great schematic that was designed by Kristen Amori, who's a stock assessment scientist here in Juneau. And she was highlighting sort of the different parts of this process. So I'm going to walk through those for a minute. First of all, there's communication at multiple stages. So communication among the geneticists, stock assessment scientists, and the survey team. And after quite a bit of discussion, we realized that it's actually really important what type of assessment is being addressed and the data that we're trying to generate for that assessment. So I'm going to go into more detail there in just a moment. But the parts of this include basically, your scoping, the need to identify what data actually is required, and then resource acquisition. So as one can imagine, for larger surveys, you need more resources, more funding. And for pilot studies, you might be able to piggyback on existing funding. Then you have a process of survey design and sampling design and actually operationalizing the survey. So collecting the data, generating the data, and then finally the modeling piece. And understanding who is responsible for each part of this is pretty critical, because this modeling piece, in my mind, initially was done by the stock assessors. And in reality, that might be done by a quantitative ecologist, by a geneticist, but clearly somebody needs to be able to do that final integration piece. And you need to know that it can be done before you actually spend all the rest of the money on the survey. And kind of our 30,000-foot view takeaways are that these interdisciplinary teams are key. So, really, eDNA researchers can't do this work alone, and stock assessors, don't have the eDNA or the genetic tools to be able to do it without us. And that, as usual, there's this really importance in communicating the data requirements, including levels of uncertainty. So understanding, what's required ahead of time so that we can generate relevant data. And you don't want to get to the end of the process and realize that you haven't really actually generated data that's useful for the management goal. Okay, and I said I was going to kind of return to why the assessment type matters. And one key reason is because there's different resource availability, i.e. money, for different species. And that means that, these species have dedicated surveys, or it's data collected from multi-species surveys, or it's more opportunistic sampling. But that's going to directly influence the ability for eDNA to also get resources for those species. And then there are opportunities for eDNA to contribute in different ways, depending on what the data needs are for these different assessment types. So it could be something like species ID, and that could, be affecting any one of these assessment types. But abundance and biomass trends, might be something that's pretty well covered by a different data stream in a data-rich species. And eDNA might be able to contribute something like, information about a different life stage based on its different selectivity as a different gear type. So, again, this is a piece that requires communication and an understanding of what actually goes into the assessment before sort of suggesting that eDNA can contribute something really novel. And then sort of just providing a little bit more clarity around examples here. So for data rich, eDNA indices might be standalone surveys. So similar to the Pacific hake example, you basically have the entire assessment unit or the stock sampled by eDNA, and then that eDNA index can operate independently in the stock assessment. And that's probably typical for age-structured models where you can sample the entire unit. Whereas for data-limited assessments, eDNA might form the basis of an assessment, because for data-limited species, there are very few resources available, and so surveys might typically only partially sample the population. Data might be garnered from multi-species surveys, and opportunistic surveys can fill data gaps. And so, eDNA might have more opportunities to contribute sort of with fewer resources for these data-limited species. So, sort of to reiterate what I just said, the data-limited applications for eDNA are often less resource intensive, and particularly in current funding environments, this could be a really good place to put some energy. Okay, so that was kind of the framework that we came up with. And now I want to pivot just a little bit and show some pilot data for a data moderate, data limited management application. And these data were collected and generated through sort of a number of folks at the Alaska Fisheries Science Center within the genetics program, and then also with our survey group. And one of the questions that I know occurs for a number of management agencies, but is certainly on the mind of folks at the Alaska Center, is whether species that inhabit rocky habitats where you can't easily pull trawl nets are undercounted by fishery independent surveys that are trawl-based. So if it's untrawlable habitat and they're never trawling there, are you missing a suite of species that primarily live in those rocky habitats? And so our pilot was in the Gulf of Alaska, and of particular interest is what we use in management terms, what we call the other rockfish complex. So it's a series of 27 non-target species of rockfishes, and they're assessed as a single assessment. And some of that might be because they're cryptic, and so identification is not straightforward, and some of that might be because they're not that abundant, and so they don't warrant sort of their own individual assessment. But this is definitely more of a data limited complex where eDNA could potentially be particularly interesting. And then the other relevant thing is that they're non-target species. So the assessment is not providing a fishing cap with fishing on these species as a goal. It's rather providing a cap of potential bycatch when fisheries are targeting other species that might co-occur. So there's like a very clear management application. There's a question. The fishery science centers have been trying to address this type of question with other sort of novel gear like cameras. and eDNA has an opportunity to provide some additional insight. So we went out on the Gulf of Alaska bottom trawl in 2023, and the water sampling was paired with a drop camera. And so there's video data that hopefully at some point in the near future we'll have an opportunity to collaboratively analyze. But there were just a small number of stations for this pilot, so there were seven trawlable and 8 untrawlable stations. And there was also a technical eDNA requirement. So this is sort of project development work that was done by Kim Ledger, who's a geneticist in our lab group. And what we realized is that actually in order to identify the species of rockfishes in this habitat, we needed a new metabarcoding primer set that's specific to Alaska rockfishes. So the existing primer set was developed in California and didn't have kind of the resolution that we needed for Alaska. And so Kim did this great work with tissue samples of 42 rockfish species from the Alaska current, so sort of British Columbia north, and developed this new primer set that was going to be usable for addressing this management question. One important note, so for folks who know rockfishes, they are always challenging in terms of species issues. And in the primer region, so the sequence of DNA, it's unique to most, but not all species of rockfishes. So what I'm showing here is a haplotype network for dark, northern, and dusky rockfishes. And what you're seeing with the shared colors in those pies is the same haplotype sequence that's occurring across multiple of these species. So, we can do a lot better for species ID than the previous primer set, but there are still a few species groups that cause some species ID issues, of course, because rockfish. So from these trawlable and untrawlable sites, what we detected were 15 species of rockfishes. And what you're seeing in these bars is basically non-overlap, with just a couple exceptions, of the species found in trawlable and the species found in untrawlable habitats. So it's just sharp chin rockfish and Pacific Ocean perch that are occurring in both types of habitats. So that's, again, small sample size, relatively few stations, but really compelling initial pilot data. And looking at this ordination plot, where the colored dots represent the stations, and then the black dots that are labeled are the species of rockfishes associated, with different bottom types. You have also sort of this clear difference in community of rockfish between the trawlable and untrawlable habitats. So again, pretty compelling that when you're trawling and you're not sampling that rocky habitat, you're missing a whole rockfish community. And in terms of pilot data, this is suggesting that eDNA surveys could increase rockfish observations in that untrawlable habitat. So really promising pilot data, and our next steps are to discuss these results with the survey team and sort of scale up what's the next part of this process. Okay, so this is a lot of text, but this is actually the roadmap that Dan initially scoped. So stock assessment scientist, right? Creating a roadmap for integrating eDNA into assessments. And what I've circled is where this pilot study falls in that process. So it's basically at the top. So we have this whole sort of flow chart of next steps of communicating between geneticists and stock assessors and survey technicians, and iterating through the next parts of this process to actually integrate eDNA into an assessment. So encouraging pilot data, but still a long ways to go before we're actually talking about this in terms of assessments. And then I also wanted to highlight sort of the future of eDNA for assessments. So the field of eDNA is, obviously just like rapidly growing, and there's a lot of interest. And so for assessments, it seems like the most obvious next steps will be more of these paired surveys, particularly camera and acoustics, and allowing those paired surveys to increase the spatial coverage and potentially decrease the cost of the survey if eDNA can be incorporated in a complementary way. Using eDNA on autonomous sampling platforms. So there are some groups that are doing a ton of R&D in this realm, putting eDNA sampling on sail drone, on moorings, on AUVs, and allowing eDNA then to be collected in different environments. Again, how that exactly works in the assessment needs to be figured out with the assessment scientists and where that data need exists. So maybe it's a different time of year than where normally the assessment surveys occur, or higher frequency of sampling, but definitely some opportunities for autonomous sampling to complement existing survey data streams. There's life stage information that we're just beginning to be able to glean from eDNA and its complement eRNA. And to the extent that folks are able to actually use epigenetic aging with eDNA, it'll be really beneficial to stock assessments, because for a lot of these data-rich stock assessments, you have age-specific information. But I think in these more technical elements of eDNA, it's going to be really important for geneticists to communicate around what eRNA and what epigenetic aging can get you, and the complexity of how actually we can generate those data, and generate those data from sort of wild samples. And then finally, there's a lot of interest in how to figure out population genetics or stock structure from eDNA. If you could, rather than collecting fish and then sort of performing traditional population genetic analyses on samples, you could just sample water and get that same information. That would be sort of a boon for a lot of traditional fisheries stock assessments. But the mechanism to actually do that from the eDNA sampling and analysis side has not been fully fleshed out. So that, I'm sure, we'll see more developments in the future. Okay, and I think I raced through that basically, but I want to thank a lot of the collaborators that I didn't previously mention. So there have been a ton of sort of data collaborations and conversations with a number of folks, a lot of whom have helped me to understand more of how assessments work and what the data structures look like, and how eDNA can play into that. And then, of course, the genetics program at the OCBA labs, and the support that the folks who are generating data and making sure that all of our supplies are stocked can provide. So with that, I'm happy to take questions and or happy to respond to any email questions that folks have after the fact. Thank you.
Thanks, Diana, for this wonderful presentation on how we can begin to incorporate eDNA into stock assessments and fisheries management. Scaling eDNA is one of our highest challenges at NOAA, and this is a very good start. Before we have a few questions coming in, and we'll wait for a few more, but a question I have is if you could go to the slide with Dan's table that outlined the major steps to integrating eDNA, and share a little bit more about what's next.
Yeah, great question. So essentially what we were doing with the Gulf of Alaska study was, a validation or a pilot study. And so, we literally have a meeting on the calendar to share those results with the folks from the survey team that participated in those data collection, and then integrate the information from the camera data. I think one of the things that I'm increasingly finding sometimes when I'm comparing eDNA to other data types is that I assumed that there was more information being garnered from those other data types than there actually was. And so sometimes the eDNA is just like way more information than what folks were operating with previously. So this, has happened with diet data and some other data sources. So I think, it's not clear exactly what to make of that integration. But I think with just the eDNA, outside of that, the next step will be from the survey team talking to the stock assessors. So, fortunately, Kristen Amori, who participated in building this roadmap, actually does the stock assessment for the other rockfish complex. So we already have a year of experience sort of going through this process of figuring out how to sort of speak less jargon or less of our own jargon, more of the other person's jargon. And so discussing how an eDNA index or how these data from eDNA could be used in her assessment is like, I think it's going to be much easier to have that conversation because of all the work that we've put in already. Mm-hmm. And then I think the other piece that is non-trivial is identifying, how do you get funding to do this work? A pilot study with 15 stations, that you can sort of do that without a lot of dedicated funding, but to do a large scale full Gulf of Alaska study, especially if you're going to additional habitats that you're otherwise not sampling, that becomes much more costly. So where do those funds come from? Where do those sea days come from? So there are lots of considerations outside of kind of our standard lab operations that become things that, hopefully the geneticists in conjunction with the managers and the survey technicians can kind of address.
Most definitely. I mean, these surveys are so cross-functional, and eDNA and trawling is, I'd imagine, just a single part of them. And so effective integration is just as important as integration, right?
Yes.
So our first question is from Shanara, who asks, why did you choose rockfish? Was it a suggestion from a management or scientific or scientist?
For the pilot study, we looked at rockfish in part because the question of trawlable and untrawlable habitat and whether we're sort of underrepresenting species in untrawlable habitat has a lot to do with rockfish, because rockfish are a lot of some of the species composition in those locations. It's also just, in our part of the world, in the Gulf of Alaska, there are a lot of different rockfish species. And so that's, no small part of what we think about in terms of fisheries management. And then, I guess in some ways it's because it's more in our genetic species sweet spot. So I've done a lot of work with rockfishes. Our lab group has done a lot of sort of population genetic work with rockfishes. So it seemed kind of like it was meeting a lot of goals. Those same samples have been analyzed for fishes more generally. I just didn't show those data. So, the species composition also separates out using like a more general fish primer, like MyFish is what we use. So, so we do have those data. This is one of the classic benefits of eDNA samples, is that you can look at those samples first for one species group of interest, and then you can look at them again for a different species of interest.
Thanks, Diana, for that one. Our next question is from Sarah, who notes that eDNA measures both what was there and what is there, and asks, is there a way to differentiate between past and present when sampling eDNA?
So this is a great question. So I focused on sort of our roadmap for integrating eDNA into assessments. The other part of the paper that we wrote, essentially, is dealing with what we've identified as the key challenges or perceived challenges in actually doing, sort of the integration. So convincing people that it's worthwhile to use eDNA despite the fact that it's sampling things that both are there and were there. And the spatial scale of eDNA is one of those key challenges. I don't think, and this is, maybe in part a personal opinion, but for the spatial scale of assessments, so when your sampling locations are, tens of kilometers, maybe hundreds of kilometers apart, that's not the spatial scale for which we're worrying about eDNA sort of persisting or moving around. And so what we concluded sort of with our group is it's okay to think about eDNA the same way that you would think about a trawl net, where what you're sampling is functionally there, because you're not double counting. You're not counting a rockfish that was on one side of the Gulf of Alaska and then counting it on the other side of the Gulf of Alaska. So, that might not be true for every assessment, but for a lot of these commercial marine species, the spatial scale and sort of the persistence or the movement of eDNA is just not a problem given the way that these assessments are conducted.
Awesome. Thanks, Diana, for that one. Quite a few questions coming in, so we'll move right along. This next one is from Hassan, who asks, what is the typical time frame from collecting an eDNA sample in the field to obtaining its results? And what are key factors that influence this timeline? And I think this question can relate to, right, eDNA can sometimes be a, quote, faster way to identify large sets of stock and fisheries data. So given the time it takes to process versus trawling and counting, maybe you could talk a little bit about the timeline differences and how eDNA may be comparable as a faster method to integrate into stock assessments. Does that make sense?
Yeah, no, and this is a question I've received from stock assessment scientists, particularly because of the usually annual timelines for stock assessments. Mm-hmm. And so this is a great question, because if we collect samples and it takes 3 years to get the data, that's not useful to a stock assessment. But everything that we've done so far has been, relatively small scale with very little support. And so if you think about actually providing an eDNA survey with the resources, both in the field and then also in the lab, to generate data at the time scale required, what I've told our assessment group is that, we could do all of the data generation within, a month, 6 weeks, depending on the number of samples, if we had the resources to do it. If we had the technicians to actually extract those samples and run those PCRs and analyze those data. And so, I think there are examples in fishery science, certainly for the work that we do for salmon bycatch, where, if you have a timeline and you're funded to actually generate data on that timeline, then you can do it. If you're unfunded, it's really hard to generate data on a timeline because you're constantly going to be competing with time for technicians. So I guess that it also depends on scale.
Scaling has a huge dependence on time frame.
Yeah.
Alrighty. Our next question is from Hannah, who asks, could you elaborate on the potential effects of currents when sampling eDNA?
Yeah, so this sort of goes back to the question about basically treating the eDNA that you're collecting as though you're not double sampling it somewhere else. And I think it, I just have to reiterate that if you're worrying about movement of eDNA at a really small spatial scale, then this might not be the appropriate assumption. But for most of our assessments, that's not the case. And so the idea that, for example, in an acoustic survey, you're also not removing those fish with a trawl net. And so in theory, you could be resampling that, that fish somewhere else, but they don't really, they make the assumption that they're not doing that. And so there are a lot of these parallels with sort of the practical implementation of the survey that I think we can learn from the approach that was used with adopting acoustic surveys. In terms of the currents, for all the data that I've seen, most of the DNA that we're sampling in a given location are, that DNA is generated by the fish that are like really proximate to that location. So dispersal of individual DNA particles beyond, a kilometer would be, certainly possible, but probably not the dominant signal of what you're seeing in a given area.
Awesome. Thanks, Diana, for that response. Let's pick up another question. This one is from Jaden, who asks, when it comes to rockfish, how reliable is eDNA sampling techniques at distinguishing between species? And I'm sure Diana can speak to this, but Jaden, I would recommend checking out our NOAA Omics YouTube page, which has a great presentation by Anita Ray on this exact subject. But Diana, would you like to touch on it real quick?
Sure. So I mean, there's a reason that we needed to develop a new primer set to distinguish rockfish species. And then essentially for that suite of species that we essentially couldn't distinguish fully, so the northerns, the darks, and the duskies, we actually dug into the mitogenomes, so the mitochondrial genomes that were kind of left on the cutting room floor from a whole bunch of low coverage whole genome sequences that we generated for those species, and so that we could basically scale up from the, let's say, dozen samples or so of tissue that Kim had initially used for that analysis. And so now we had 200 samples that we could analyze full mitogenomes, and there's just not a lot of sequence to distinguish some species of rockfishes. So it depends on the species that you're looking for. Like, out of the 42 species, I think there were fewer than 5 maybe that had these species complexes. And even then, there are some haplotypes that are unique and some that are not unique. So in that haplotype network, some of those haplotypes for northern rockfishes are unique to northern rockfishes, to the best of our knowledge. So based on the sample of 200 or some odd species or some odd samples, but then there were a few haplotypes that were not unique. So, we can have that level of granularity in our analysis, and that can happen, well before those data are shared with stock assessment scientists, of course, right? So there's still the same level of genetic analysis and diligence around making sure that we're very careful about what we're confident in and what level of uncertainty we're accepting.
Awesome, thank you, Diana. And we're at time, so we'll just take one more question and conclude today. This one's from Alberta, who asks, how do you think the use of eDNA in stock assessments, for example, as an additional index, should be introduced to fisheries management councils?
Good question. Okay, so what's nice about my position is that the Northwest Center is going first. So, they have the Pacific hake example, and they developed an index. They invested a huge amount of resources in collecting all of those data and analyzing them. And they actually incorporated the eDNA index as a sensitivity run in their stock assessment this January. So, so it's been done, which makes everyone who comes next, it makes it just a little bit easier on us, right? Because as I can point to the hake example in my presentation, our stock assessment scientists at the Alaska Center can point to that example as a way that this can actually be done and be operationalized. In terms of communicating with the management councils directly, one of the things that we've been communicating about within the Alaska Fisheries Science Center is sort of acquainting folks with eDNA ahead of time. So in kind of like a hot topic for I think it was the Bering Sea ecosystem report, we added a little description of some of the eDNA samples that we've been collecting in the Bering Sea. And it's, we don't have the full scale data set. It's not a time series that's been analyzed for a stock index at this point, but basically just familiarizing folks in the management council process that we are collecting eDNA, sort of like stay tuned. It's Kim Ledger and myself who are, doing most of this work for the Alaska Center. And so if they have questions or comments, sort of reach out to us. So I think it's developing that familiarity and then maybe probably sharing some of this pilot data. For data to be incorporated into a stock assessment, it really has to go through the ringer, and eDNA, has not done that for the North Pacific Fisheries Management Council yet, but I think that we'll probably have some reasonable data to share with them, in the near future.
And the key to that is yet. Diana, her team at the Alaska Fisheries Science Center and NOAA Omics is definitely leading the way and doing our best to understand more about how eDNA and omics can fit into fisheries management and especially stock assessments. So with that, Diana, thank you very much for presenting today. This was a wonderful, very informative and cross-communicative presentation to omics researchers and fisheries stock assessment scientists alike. And thank you all for joining us today. Our next seminar series will be on May 21st at 2 p.m. ET. More details to come with that as we approach that date. We hope to see you there. Thanks for joining today, and thank you, Diana, very much.
Thanks so much for the opportunity.